Hello everyone, this is a B.Sc third year semester 5th. Today I have to discuss the unit number 3, the another aspect that is the synapse from unit number 3. Synapse. Uh, Sir Charles Sherrington was the first person who applied the term synapse to the junction point between the two neurons. दोन न्यूरॉन मधला जो जंक्शन पॉइंट आहे त्याच म्हणजे दोन न्यूरॉन ज्यावेळेस एकमेकाला जॉईंट होतात तो जो पॉइंट आहे त्या पॉइंटला सिनॅप्स सर चार्ल्स शर्जिन शर्जिननं स्टेटमेंट केलेलं आहे त्या टर्मला सिनॅप म्हटलं जाते. ओके. The physiologically importance of synapse for transmission of nerve impulses was established by मॅकलिनॉन इन 1963. 1963 साली व सिनॅप्सचं इम्पॉर्टन्स काय आहे फिजिओलॉजिकली की ते नर्व इम्पल्सेस करते फ्रॉम वन टू अनदर. नर्वचे जे इम्पल्सेस जे आहे ते इम्पल्सेस तो घडून आणते असं मॅकलिनॉननं 1963 साली म्हटलं आहे. तर देर आर टाइप्स ऑफ सिनॅप्स. एक्झोडेंट्रिक सिनॅप्स. The synapse develop between the axon of one neuron and dendron of another neuron. म्हणजे axon एका न्यूरॉनचं आणि dendron दुसऱ्या न्यूरॉनचं यांच्यामध्ये जे सिनॅप्स किंवा यांच्यामध्ये जे जॉईनिंग होते यांच्यामध्ये जे अटॅचमेंट होते त्याला म्हटलं ते एक्झोडेंट्रिक सिनॅप्स. एक्झिनिक सिनॅप्स सेकंड टाइप. The synapse develop between the axon of different neuron. दोन वेगवेगळ्या न्यूरॉन मधला axon मध्ये जे संबंध आहे that is also called as exogenic synapse. Axosomatic synapse means काय? In the cerebellum of the axon, basket cells make synapse with the pattern of the Purkinje cell. Okay. Then dendrodendritic synapse, the synapse which develop between the dendrites of different neurons. वेगवेगळ्या न्यूरॉन मध्ये सिनॅप्स घडून येतील न्यूरॉनमध्ये. पुन्हा dendrodendritic synapse. Somosomatic synapse is the most primitive types of synapse which develop between the cytons of different neurons. There are again two categories of synapse, chemically transmitting and the electric transmitting. Electrical synapse will be considered first since they behave more simply. An electrical junction between two neurons, also local circuit current from an action potential in one to spread into the other and depolarize. Structure of synapse, then chemical synapse. Under electron microscope it is seen that presynaptic fibers end is an expanded terminal. The synaptic node and soma of postsynaptic cell each has an intact membrane. The membrane of the synaptic node known as presynaptic membrane and the membrane of axon or dendrites of the postsynaptic cell is known as postsynaptic membrane. The synaptic knob has got initiate the contact with the portion of the membrane of the postsynaptic cell. The synaptic knob are separated from the soma of the postsynaptic cell by the synaptic cleft. The synaptic cleft has an average width of approximately 20 nanometers. The synaptic cleft actually represents the real discontinuity of cell cytoplasm in the synaptic junction. In the synaptic cleft of brain cortex, the presence of parallel intersynaptic filament of about 50 nm crossing the synaptic cleft. These filaments are fixed at the both ends with presynaptic and postsynaptic membrane. Beside there is a web of filament which is implemented on the postsynaptic membrane and extended at a varying and considerable distance in the postsynaptic cytoplasm. This web of filament known as subsynaptic web. This synaptic web has observed in the central synapses of the brain cortex and the retinal synapses. The synaptic node contain mitochondrial and a large number of synaptic vesicles. Means it contain mitochondria and large number of synaptic vesicles about 3 lakh of which are normally in the terminals of the axon. The synaptic vesicles are more concentrated toward the site of the in fronting the synaptic cleft. The synaptic vesicle contain the excitatory transmitter material that mediated transmission of impulses from the pre-sympathetic neuron to the post-sympathetic neurons. It is claimed that the mitochondria probably taken part in the synaptic of transmitter substance on the post-synaptic membrane. The hypothetical receptors are present which play an important role in the permeability of membrane. In the ring of synaptic inhibitors, the synaptic enzyme is present. As for example, acetylcholinesterase in the cholinerase junction, which is capable to destroying the acetylcholine. This AMI structure of synapse. It show that mitochondria here. This is the presynaptic membrane. This is the postsynaptic membrane. Here vacuole, the membrane, presynaptic. This AMI structure of synapse. Then synaptic transmission. The transfer of nerve impulses across the synaptic junction is known as synaptic transmission. म्हणजे ट्रान्सफर जे होते nerve impulses करून across the synaptic junction known as synaptic transmission. The process is believed to be brought by the release of chemical substance at the synapse and taken place in following step. Release of neurotransmitter substance. The neurotransmitter substance synthesized in the terminal process of the axon are stored in the synaptic vesicles. On the arrival of an nerve action potential through the axon into the terminal knob by some unknown mechanism, the vesicles release the neurotransmitter substance. Calcium ion required for the release and magnesium inhibit the process. After the release of the transmitter, the vesicular membrane moves into the cytoplasm and is used package new transmitter substance into that. Diffusion of the transmitter substances. Neurotransmitter substances released by the pre-sympathetic terminal diffuses across the synaptic cleft and bind to the specific receptor site on the subsynaptic membrane. In the vertebrate neuromuscular junction, acetylcholine is released from 100 to 30 presynaptic site and diffuses across a distance of less than 1 micrometers. Permeability alternation of the here permeability alternation in the subsynaptic membrane, the binding of the neurotransmitter to the receptor molecule is accomplished by alternation in the permeability of the subsynaptic membrane. Two types of alternation in the permeability are observed. The first is generated type, in which the permeability of the post synaptic membrane to all type of ion, bringing about a depolarization of the membrane and excitatory post synaptic potential is produced. The second type increase the permeability of the membrane to potassium positive and chloride ion, causing hyperpolarization of the membrane and inhibitory post synaptic potential. If the synaptic potential are great enough to produce them sufficiently strong local current, a spike is generated in the approximate region of the postsynaptic neurons. म्हणजे जर समजा synaptic potential is great to produce the sufficient strong local current, at that time the spike is generated in the appropriate region of the postsynaptic neuron. Destruction of the neurotransmitter substances. Here the neurotransmitter substances is destroyed quickly, so the normal subsynaptic resting potential are restored and neuron may responded to a new stimuli. the new stimuli, action potential arriving to the nerve ending result from the ion current, calcium entry into the presynaptic ending, liberation of acetylcholine molecule contained in the synthetic result by fusion with membrane and opening into the synaptic cleft, diffusion of neurotransmitter within the cleft, interaction with the receptors, opening of the postsynaptic channels, sodium and potassium current through the channels, interaction of acetylcholine with acetylcholine esterases and hydrolysis take place. Choline is take up again the post sympathetic re-potential. This the diagram shows that the component of the acetylcholine system and step in the mechanism of neurotransmission. It's all about the synapse. Thank you.